In order to understand the alternate approach, we have to understand that this alternate approach was building on top of what biologists, neurobiologists in particular, had discovered until that point. And again, we're talking about roughly the 1950s when the Dartmouth conference happened, because there were people who attended the conference who were not in favor of that logic based approach. They had a different approach in mind. And that approach was inspired more by what neurobiologists had uncovered until that point about the structure of the brain and how the brain works. So, just to give you a brief background into what the state of knowledge was, the relevant knowledge about how the brain works. So, in the 1600s, Robert Hooke, who was a contemporary of Newton, had improved the design of the microscope, which had been recently invented, in order to see different things. He looked at a lot of different insects and all kinds of objects under the microscope. And when we looked at a thin slice of cork, he saw that there were small compartments inside the cork, which he called as cells. And then later, Schleiden and Schwann, two independent scientists, eventually saw that these cells are not just inside cork, but they are inside virtually all plants, and in fact all animals as well. So they developed, or they proposed, what was known as the cell theory of life, which was that all living organisms are composed of cells as basic units. And that was just based on observations of different specimens of different body parts of plants and animals under the microscope. Now, Camillo Golgi was an Italian biologist who developed a staining method. So these staining methods were helpful in identifying the internal structure of a cell, because depending on what chemicals you stained a cell with, it could stain different parts of a cell with different colors. Right. So that exposed the internal structure of a cell and eventually led to the discovery of those different cell organelles and the genetic material, DNA, RNA, and so on. So Golgi developed a staining method which was particularly helpful in revealing the structure of cells in the nervous system, in the brain. So he added a solution of a chemical like silver nitrate to the brain tissue, and when he did that, a chemical reaction occurred which caused a black stain to accumulate within the brain cells, right, the brain cells in that slice. And so when he looked at those cells under the microscope after staining, he saw something like this. This is actually a diagram from that he published in one of his books. He saw that inside the brain there seemed to be cells, but they seem to be forming a very complex network, which he at that time called as a reticulum. So he proposed that the nervous system, which includes of course the brain, but also the spinal cord and other parts of the body where these wire-like projections go, and they seem to influence a lot of things, including the movement of muscles. So he proposed that this nervous system is a spider web, is like a spider web of wiry projections, wiry, you can say, tangles of continuous extensions. Of brain cells. So somehow the brain cells are able to grow these wires or wiry projections that seem to join with the projections of other brain cells and fuse into some kind of continuous network. Now, building on the staining method that Golgi developed, Santiago Kahal, the Spanish biologist, uh, in fact, he perfected the staining method even more, and he looked even more carefully at these networks, at this so-called reticulum, and he saw that it was not a continuous network. There were gaps between the projections from one of these cells and the projections from another cell. Okay, so what he found, in fact, let me show you the diagram first. This is one of the diagrams he published. What he saw is that the projections from, say, one of these cells, say this cell that is labeled F, you can see that the projections end here, and there is a gap between the projections of this out of this cell and the projections out of this other cell. Right? They don't touch each other, and so he disagreed with Golgi on this reticulum being continuous. He said that there are individual uh, discrete cells that make up the nervous system, not like a continuous network. These discrete cells became known as neurons, and they are not continuous, but they are contiguous with each other, separated by intervening space, which was later called or named as a synapse. The synapse is the gap between the projections of one neuron and um, the neighboring neuron. So the neuron was the name given to a cell in the nervous system. All right. So uh, now there were other biologists like, for example, Galvani, Luigi Galvani in Italy, who had figured out that the way a neuron uh, influences or communicates the message down to different muscles, making those muscles move, is through an electrical mechanism. Because he was able to do these experiments where dead frogs that were completely dead, he could make their muscles twitch by sending small bolts of electricity down their spinal cord even after they had died. And so um, you can just add that here, that Galvani had shown that the mechanism by which neurons, by the way, a bundle of neurons uh, that go down any particular part of the body is called a nerve. A nerve is just a thick bundle of individual neurons. So the mechanism by which neurons send messages is electrical. Some kind of electrical signal or current goes down a neuron. The next people till that time were thinking that maybe there is some kind of fluid or gas that uh, flows down a neuron. So the neuron could be like a pipe through which some fluid flows. But Galvani pretty much settled that question and established that it's basically electricity. Now, through the microscope, through these staining methods, people figured out that a generic neuron has the following structure. Near its head, it has these tree-like projections, which are called as dendrites. So the word dendron means the tree, and so dendrites are these projections. And there are on an average of, say, 10,000 of these projections near the head of a nerve cell. Now, inside the main body of the cell is the nucleus, which contains the genetic material. The main body is called the soma. Soma means body. And then we have um, um, a long cable coming out of the cell body, which terminates again at a bunch of projections. This long cable is called an axon. And the endpoint of the axon where these projections are, these so these projections at the end, they are called as axon terminals. Axon terminals. So on average, you can think of a neuron as having, say, around 10,000 dendrites and around 10,000 axon terminals. And these axon terminals are very close to the dendrites of other neurons that are in the neighborhood. But there is space between those terminals. They don't literally touch each other, as Santiago Kahal had found. Now, different neurons are of different lengths. There can be neurons that are less than a millimeter to, say, neurons that are several feet. In fact, the largest neurons would be down the spine of a blue whale, so almost 60 feet long. And biologists that time found it easy to understand the structure of a neuron by studying squids. So squids seem to have neurons that are so thick that they can be seen with the naked eye. And so research on squids, the nervous system of a squid, is what led to the discovery of a number of mechanisms of neuronal communication, because you could literally, plant an electrode inside, say, the axon, send a bolt of electricity, and see what happens. And by doing experiments like that with electrodes, biologists figured out that the dendrite receives information from neighboring neurons, and under certain conditions, the information it receives causes a bolt of, an electrical signal to go down the axon to the axon terminals, and somehow that message gets passed on to the neighboring neurons. Because they were able to do these experiments where you plant electrodes in the axon, send a bolt of electricity here, and you see that the neighboring neurons are going to respond to it in some way, which I'll just describe. So, let's look at, so this is the structure of a single neuron, but let's look at how a neuron acts. Broadly speaking, there are two states that a neuron can be in. The resting state is called the resting potential, and this is the state in which it's not doing anything. So in the resting potential, what happens is that the neuron maintains a negative charge inside the axon and a positive charge, overall net positive charge outside the axon. So this is a section of the axon. And so there are negative ions, an excess of negative ions inside the axon, and an excess of positive ions. These could be sodium ions or something like that, just outside. And so you can see that there is a voltage difference between the outside and the inside. And this is maintained. See, normally when you have a voltage difference, you would expect the charges to come inside and neutralize each other. But there are ion channels. Let's call them selectively permeable. Selectively permeable means a path that allows only certain molecules to go through and not others. So there are selectively permeable ion channels inside the cell membrane of the neuron, which allow only certain ions to pass through and not other ions. And that's how the cell makes sure that unwanted chemicals stay outside, and only certain chemicals are allowed to go in and go out. Kind of like how a castle is guarded by gates, allowing only certain people to come in and go out. And so the neuron actually uses energy to maintain this voltage difference in the resting potential. So it's actively pumping, you can say, positively charged ions out so that this voltage difference is maintained. And that's called the resting potential. Okay, so a lot of energy needed just to stay quiet. Just to stay quiet and maintain the voltage difference. But what can happen is that when the dendrites receive messages from neighboring neurons, there is some biochemical pathway that could get triggered from the different dendrites. And the incoming message in some form, we'll talk about that a little more in detail later, but in some form, these messages travel to this part of the neuron. This is called the axon hillock, which is an important site where something happens. Namely, the messages from different parts, from different dendrites, are integrated together. So you can think of them as uh, adding together the messages. So what are these messages? So these messages could be um, a flow of ions again, right? When the dendrite receives a message, some biochemical pathway gets triggered, which leads to the flow of certain ions from there. And so when you have ions from different parts flowing together to reach the axon hillock, think of the analogy of a water wave. So receiving a message is analogous to dropping a pebble in a pond. So that can lead to a wave, a flow of ions flowing out away from that site like ripples. And those ripples obviously get smaller and smaller as the distance increases. But assuming that under certain conditions that ripples from different parts are able to reach the axon hillock and get added together, that could create a wave that is tall enough to cross the axon hillock. So the reason it's called a hillock is you can visually think of it as a small hill or as a wall with a certain height. And if the signals received from different dendrites add up to a height that is more than the wall, then the water is going to jump over the wall. In other words, the signal is going to travel past the axon hillock. And when that happens, the ion channels, the selectively permeable ion channels, which are like gates at the entrance of a fort or a castle, those gates suddenly get thrown open. So these ions, they effectively end up opening the gate here. Opening the gate here leads to all those positive charges coming in, and they go downstream and open these gates, the gates further downstream, which again leads to the positive charges there coming in, which again go downstream, open the gates here. And so suddenly you have this positive feedback mechanism by which the charges come in, they open the ion channels um, further down. And so there is like a wave of positive charges coming into the axon, neutralizing this voltage difference. And that happens really fast. And effectively, that is the signal that is going down the axon towards the axon terminals. It's basically these positive charges streaming in and uh, changing the selective permeability of the ion channels, causing more of them to enter. Like it's like when an enemy enters into the castle, a few people enter the castle, they'll go start opening the other gates so that more enemies can come in, and they will open the other gates and so on. So as long as the signal, even if the signal is a little bit taller than the axon hillock, this positive feedback mechanism is triggered, which causes a large signal to rush down the axon to the axon terminals. And when this signal reaches the axon terminals, what it does is it discharges. So there at the axon terminals. So think of one of the axon terminals. Okay, let's zoom inside one of these axon terminals and what is happening there. So this part is one of those axon terminals. At those axon terminals, there are small balloons, we call them as vesicles, which contain a chemical. Uh, so think of it as a balloon loaded with some cargo. And when these ions come in, the message comes in, they cause this vesicle to discharge the cargo. So in other words, the vesicle fuses with the cell membrane and releases the cargo inside. It's a chemical called a neurotransmitter. And it's released into this river or the extracellular space between the axon terminal of one neuron and the dendrite of a neighboring neuron. So these neurotransmitters are released into the synapse, and they travel across the synapse, and they bind to receptor molecules in the dendrite of the neighboring neuron, similar to how a key binds to a lock. So the neurotransmitters have a certain shape, they have a certain charge, and the receptors in the cell membrane of the dendrite have a complementary shape, maybe a complementary charge. So the binding of the neurotransmitter to the dendrite triggers a new wave, which you can see here, from those terminals. And so once again, this wave goes down the dendrite towards the cell body, and at the same time there are obviously around 10,000 dendrites. So all of those in parallel would be potentially receiving signals, or maybe not all of them would be receiving, right? Maybe only some of them would be receiving. But the signals from all of those dendrites have to be integrated together again at the axon hillock of the next neuron. And again, if the net sum of all those signals exceeds the threshold of the axon hillock, then a message is sent down the axon of that other neuron. So that other neuron would suddenly become active and shoot down an electrical signal to its axon terminals, and the same process then continues. So a neuron can be in this resting potential state, but it can also transition into a more active state. We call that the action potential, which is the state when the signals from the dendrites end up exceeding the threshold of the axon hillock. So in the action potential state, what happens is a wave of excitation, you can say, crosses the axon hillock and sets off positive feedback mechanism for shooting an electrical signal down the axon to the axon terminals. So wave of excitation, that's a phrase you will also see elsewhere. So I hope these terms are clear. Resting potential, the default resting state of a neuron. Action potential, a different kind of state where the voltage difference that it has, that is using a lot of energy to maintain, is suddenly disrupted by this wave of excitation that causes the sodium ions from outside to come in and open up even more channels further down the axon, and then that's the electrical signal we're talking about. And then once that happens, once the neurotransmitters, so at the axon terminals, neurotransmitters are discharged into the synapse, released into the synapse. And after that, the neuron will work again to rebuild that voltage difference, go back to the resting potential state, and wait for the next signal. Now, this is the condition. The crossing the axon hillock is necessary. If you don't cross the axon hillock, then any signals coming from the dendrites are not going to trigger an action potential. The neuron will continue to stay in the resting potential state. And this crossing the axon hillock, you can literally think of it as adding an analog signal, adding analog signals from all the dendrites. So the signals from all the dendrites, think of them as values that are being added, and once those values exceed their threshold for the axon hillock, this action potential is set off. And this electrical signal, you can think of it as digital, because it doesn't matter by what quantity you exceed the axon hillock. If you exceed it even by a small amount, the same kind of signal is going to be sent down, because basically the floodgates would be opened regardless. So the input is kind of analog, but the output is digital. So what happens in the body is that there are dendrites in the eye which receive signals from the external world, and those signals can then set off action potential inside some of those dendrites, and they would transmit an electrical signal to other neurons that are deeper inside the brain, and those neurons would be deeper inside, they would be part of the brain, they would be part of the spinal cord, and so there are obviously lots of neurons connected to one another there, and somehow the message passes through. Uh, and it's not just one dendrite that's receiving the message, could be lots of dendrites receiving lots of messages in parallel. They're all integrated together, and eventually maybe a signal goes down one of those nerve fibers to one of the muscles, getting that muscle to contract and thereby making you walk. Or it could even be the heart muscle, right? Getting the heart muscle to beat more rapidly, um, and so increasing the blood pressure or respiration rate, right? Maybe getting the diaphragm to contract more frequently, so making you breathe faster. So when we one of the problems with the phrase artificial intelligence is that uh, we tend to think of intelligence as something conscious, symbol manipulation, logical thinking, something we do consciously. But a lot of our brain is actually working on tasks that are very complex but unconscious. And perception, recognizing objects, faces, is one of those tasks. And then motion, moving our hands and legs in complex ways, say reaching out under a table to grab a pen. Um, Automating those tasks can be much harder than automating some of the symbol manipulation tasks, paradoxically. So, obviously, automating what's going on the perception side, recognize faces, objects, and so on, that would be part of, say, uh, today we would put that under computer vision. And then getting muscles or some kind of effectors, uh, muscles, glands, or whatever, to move around, to do whatever needs to be done, that would be today part of robotics, automating that. Okay, so let's see. Anything else that I'm missing here? Oh, yeah. Apart from neurons, there are other kinds of cells in the nervous system, and those are known as glial cells. And this word comes from the word, uh, from a word that also means glue. Because earlier people thought that neurons are the main stuff of the brain, and these other cells that are inside the brain are more like stuffing. They don't have a very crucial role, they just play a supporting role. So they're called glial cells, and they're also of different types, and now people are finding that they're actually a lot more important than what they used to think earlier. But one type of glial cells um, forms uh, something analogous to the wrapping around a wire. When we have a cable that insulates the wire inside it, the wire that actually sends the electrical signal. You can think of the glial cells here as forming an insulation around the axon, uh, which makes the signal go through the axon faster. And this insulation around the axon is called the myelin sheath. And it increases the efficiency of this transmission so much that people have actually found that during human development, as a child develops new abilities, they have found that the corresponding areas of the brain that they use for that activity is getting more and more myelinated, indicating that the myelin sheath is really important for uh, development. And in fact, they have found that in the cortex, in the frontal cortex, the myelin sheath builds as late as the early 20s. And since the frontal cortex is known to be responsible for things like self-regulation, that explains why young people might not have self-control as much as, say, an adult. And for each of these mechanisms, there are issues that can happen when the normal functions are disrupted. For example, if the myelin sheath, um, there are scenarios where the myelin sheath gets attacked by the human immune system, and that leads to a disease called multiple sclerosis. Um, Likewise, there are other disorders that can happen when this transmission is again disrupted for one reason or another. For example, if there are not enough neurotransmitters, then biologists have found that certain kinds of depression are correlated with the absence of a sufficient amount of neurotransmitters uh, in the brain. Likewise, there can be drugs that could change some of this normal functioning. Hallucinogenic drugs, for example, can imitate some of the neurotransmitters. And so, even though there is no external signal, perceptual signal for uh, an action potential in the neuron, a hallucinogenic drug could bind to the receptors of a bunch of neurons and trigger a wave of excitation and an action potential in those neurons in the absence of a perceptual input. And so, drugs like LSD uh, effectively act by simulating some of the neurotransmitters and binding to the receptors in the absence of those neurotransmitters. By the way, after binding to the receptor, a neurotransmitter could be either recycled, which means it could go back into the previous axon, or it could be, digested by an enzyme and sent into the cerebrospinal fluid and basically discharged out of the body. Just a terminology here. This is called the presynaptic neuron, because we are assuming that the message goes down from the axon to the dendrite in that direction. And this is something the biologists found by planting electrodes, and you send an electrical signal into the axon, you see that the neighboring dendrites get stimulated, wave of excitation can be triggered in the next neuron. So the flow of information is that way and not this way. So this is called the presynaptic neuron because the information is flowing like that. It first goes through this neuron, and then it goes to the next neuron, which is called the postsynaptic neuron. So that's some terminology. Okay. It's 10:30. Okay, it's 10:30. So I'll probably take a break. I'll just take a question. Rohit, Pegalapati.
Yeah. So, is there any are these arrangements of dendrites and axons? Is there a pattern that is common among all the human beings, or um, so I was trying to understand when somebody gets an external input, right? Each of the person is going to process it in a different way, right? So
exactly, yeah.
So the pattern of these arrangement of these neurons and axons, if there is a common pattern, why would that happen?
Yeah, so individual differences will be there because there will be differences in the number of these dendritic terminals or axon terminals. Even the threshold for the axon hillock might be different in different people. Some people might get triggered a lot more easily by certain inputs than other people. And this could happen again because of whatever genetic reasons. It could also be triggered by hormones, right? There, like in animals, for example, they have found that there are certain reflexes in the breeding season that get triggered in females uh, because the axon hillocks in the breeding season are regulated by those hormones, which cause females to, show certain kinds of reflexes for mating, which they otherwise wouldn't outside of the breeding season. So hormones can also change the threshold level for the axon hillock. The amount of neurotransmitters that are produced in different individuals, that could be different. And so there's obviously going to be a lot of variability across individuals in it's not just a purely digital mechanism, right? The strength of the signal that uh, is launched from here, that could be different in different individuals. The threshold for the axon hillock could be different. The number of receptors here could be different. The number of vesicles here and the amount of neurotransmitter could be different. How the neurotransmitters are recycled or whether they are thrown away, that could be different. And so there is a large variety of no two individuals will be alike in all of them.
Thank you.
Harish.
This is Akhil, actually. I don't know. I got logged in as Harish.
Oh, okay, sure.
So, do these axon hillocks have memory? Are these memory-less systems or memory like previous experiences? Would it increase the threshold? Yeah.
Yes. So far, what we've talked about does not involve any learning.
Okay.
So I will get into learning a little later, maybe in about half an hour.
Sounds good.
Yeah. We want to first understand how to model a neuron mathematically based on what we have learned, and then once we've done that, then we will ask what happens when we learn something, right? When we talk about memory or whatever, something that we have learned, what happens in the brain, and then we will go back to our mathematical model and
Sounds good.
enhance it to incorporate learning. Amit Jain.
Hi, Omkar. One question for so each neuron has multiple axons. So is there that boundary or threshold level at the boundary of each, or is it only at the head, wherein the first axion starts or connects to the neuron?
There is typically only one axon. There are maybe 10,000 thousands of axon terminals. At the end of the axon, you have these axon terminals, but there is only one axon, and so only one axon hillock.
Okay.
Yeah. Once the action potential goes down, it's going to trigger the neurotransmitters from all the axon terminals here. But there could be, again, individual differences in how much is triggered from each terminal. So we'll come back to that when we talk about learning.
Okay, thank you.
Byung-Kun?
Yeah, so in this diagram, only there's one connection, neuron A to neuron B. So
Yeah.
what about that a neuron is connected to multiple other neurons? In the case, there is like one axon per connection, or is it per dendrite?
Yeah, so typically a neuron will be connected to thousands of other neurons. So what will happen is that some of these axon terminals, and imagine 10,000 of them here. So maybe a few of them connect to the dendrites of neuron A, a few of them connect to the dendrites of neuron B, and so on. So because there are thousands of connections here, there can literally be thousands of different neurons that are fed information by a single neuron here. And likewise, each of those neurons could have 10,000 dendritic terminals that are taking inputs also from thousands of presynaptic neurons.
I see. So there is still single axon, but then multiple axon terminals connected to multiple different neurons.
Yeah, a single axon. There are some exceptions, but yeah, typically a single axon with thousands of axon terminals connected to thousands of other neurons through their dendritic terminals. So it's like a many-to-many relationship. One single neuron will send an output to thousands of other neurons, and each neuron will receive inputs potentially from thousands of other neurons, because there will be axon terminals from thousands of other neurons feeding in signals into the dendrites of a single neuron. So all of those signals have to be integrated together, and some of those signals need not be positive. They can even be negative. So coming back to, just adding one more thing to these neurotransmitters. These neurotransmitters can be excitatory, which means they send a positive signal to the dendrites of across the synapse, right? Which means making it more likely that the postsynaptic neuron will fire or will enter the action potential state. But they could also, some of the neurotransmitters are inhibitory, and neurotransmitters are also of different types. I think scientists estimate that there are around 100 different types of neurotransmitters. Some of them are inhibitory, which means when they bind to the receptors, they will actually try to block the neuron from going into the action potential state. They might send a negative signal, which means at the axon hillock, there could be positive signals coming from some dendrites and negative signals coming from other dendrites. And after summation, the net result will be decided by which of those signals dominate overall.
I see. Thank you.
Dheeraj?
Yeah, hi, Omkar. A couple of questions, right? So first, when the neuron A is connected to neuron B, the axion terminal is connected to the dendrite of neuron B, correct? That's how they communicate,
Yes.
or do they directly touch the head?
No, they don't touch the head. That was actually the controversy that was resolved by Santiago Kahal, who saw through his careful observations that there is intervening space.
Got it. Okay.
between two neurons.
So even if there is space, so does that mean that the dendrites and the axion terminals don't touch, but are close enough where they can pass the electrical signal, right?
By the way,
thing which you just
so these are chemical signals. So the neurotransmitters are chemicals, and so we can say overall the signal is electrochemical, right? Because down the axon the signal is electrical since it's basically flow of charge, but across the synapse it's purely chemical. Right, so you can say electrical, then chemical across the synapse, and then through the neuron again, ions flowing. So electrical, chemical, electrical, chemical, electrical, chemical. That's how.
Got it. And are all the dendrites like occupied, or I mean, I might be just a detail, but is it guaranteed that all the dendrites will be connected to axion terminals, or there might be some which are not connected at all?
So some might not be connected, but I think what happens in development is that there are a lot of dendrites and axon terminals that are sprouted, and then some of them get pruned, and I think there are other kinds of glial cells that come and literally prune out the terminals that are not being actively used. So I think it's an area of research that is quite active. This is work, I believe, that has just been done in the last 10 years or so, where they have tried to identify some of those microglial cells that seem to literally eat up or nibble away at those terminals of the neurons, like our immune system cells gobble up bacteria in an infection.
Okay. One more question. So when I'm forgetting the term, when we go into the axial HELOC, right? If the wave is not big enough to cross the wall, what happens to that wave, or what happens to that information is what I'm trying to ask. The electrons still flew through, but they couldn't breach the barrier. So what happens at that point, or is that information lost? Or
it's going to be lost. So there are, of course, it might have some side effects. Again, this is an active area of research, but apparently there are other slower pathways that can change how a neuron is structured or how it behaves based on these events. But we are talking about the fast pathway. That is, so there will not be any signals sent down the axon. The neuron will work again in recycling the neurotransmitters and rebuilding that voltage difference and potential difference between the inside and the outside. Of course, it was not disrupted in the axon here, but whatever the default state was here, it will go back to reinstating that.
Sure. Okay. Sounds good.
Surendranath.
Hi. This is similar to a follow-up question to just our last one. At Action HELOC, you mentioned that the threshold is little bit is above the signal will pass, right? It doesn't have to be doesn't require enough strength or something, as minimum as possible. How does, for example, you give me the scenario in the castle, so enemies come and open the gates, but they may not have enough strength to open the gate. So it means the whatever the threshold cross, it's supposed to have some weightage, right?
So I was going to discuss it after the break, but let me just put it as an equation so that becomes clear. So typically we will, we could represent the cell body and the dendrite part like this, and we could represent the axon part like that. And these are, let's say, the dendritic inputs, right, from x1 to xn as shown in this diagram. And the output, of course, it could go to a bunch of other dendrites, but it's going to be a digital output, which means 0 or 1. And the way it's decided is when you add the signals from the inputs, x1, the summation of all of those is greater than the threshold t for the axon HELOC, then the output sent down the axon is 1, else the output is 0.
So
Does that make sense? So the threshold is somewhere here in the axon hillock.
Oh, that's why you mentioned it's digital.
Yeah, that's why it's digital.
Okay, got it.
So, and because the output is digital, you can think of the inputs x1 to xn also as digital for now.
Okay.
So inputs are zeros and ones coming in from here. You add them together. If that ends up exceeding the threshold for the axon hillock, then an output of 1 will be sent to all of these. All of these y's will be 1.
And just another comment in the one of the earlier question, somebody asked if everybody receiving the information same way, why different people are. That time, I believe even the parallel information it's processing also influence the output, right? For example, when some message is going through some neuron pattern, and in the somebody parallelly process some other information at the same time.
Yeah. So this is all happening in parallel across. I mean, there are literally 100 trillion. That's the estimated 100 trillion connections in the human brain between axons and dendrites. Right. So 80 billion or so neurons. So obviously there'll be billions of them that will be potentially active at the same time when we are seeing something. So all of these signals are being sent in parallel across a huge network.
Hey, Omkar, one question I had was like, were we able to kind of simulate the connections in any of the neural networks in computer science yet? Because all the neural networks that I've seen up till now are all kind of densely connected. That is like neurons in one hidden layer are connected to all the neurons in the other hidden layer, but that seems to be fundamentally different in the way the biological neurons are connected, right?
Yeah, I mean they are also connected in layers. We'll talk about that when we get into the perceptron. We'll talk about that. But I believe that the neural network behind GPT-4 has roughly 1 trillion connections from what I've heard. So we are still like two orders of magnitude less than the complexity of the human brain, even in the largest networks that we have so far. But maybe we'll get there soon because GPT-3 had, I think, 125 billion connections.
Not just in the number of connections, right? But the secret maybe lies in how they are connected also, right?
Yes. Yeah, that's true. But as you saw here, here the neurons that are inside our sense organs would be receiving the signal. They would be sending it into the central nervous system, mainly the brain and the spinal cord, where the information would be integrated, and then there would be a signal sent to the muscles. So it's not like this. The neurons here are connected to the neurons here. There will be like a relatively long path. When I say relatively long, obviously the signal has to be transmitted really fast. If there is a predator that's attacking us, we want to react very fast. So the processing time shouldn't be long, but still, I mean, there will be a pathway through which the signal will go. It won't be like directly from here to there.
Thank you.
Patel?
Hi, Omkar. I had a question regarding the direction. The way it is described is like, let's say, dendrite neuron A is sending information to neuron B. It's sending from the axon network of A to dendrite of B. How about the reverse? I mean, here, since you said the transmitters, the neurotransmitters can send both positive or negative.
Yeah, no. So there is no. The dendrites don't send back a signal to the axon. Although I will come back to that point a little when we talk about learning. So of course there could be feedback mechanisms where the axon of a neuron could feed information back to the dendrite of an earlier presynaptic neuron. But it's not like the information would go back from the dendrite to the axon. It's always from the axon to the dendrite.
So how does it reverse? Because the nervous system is bidirectional. So does it mean like it's like one neuron?
So there can be feedback loops here where you can have one neuron or a few neurons whose axons ultimately provide feedback to the dendrites of an earlier neuron. But still, an individual connection is still one way from an axon to a dendrite. Just like a graph can have loops, right? I mean, you can have this neuron sending an output to this neuron, sends an output to this neuron, which could send an output back to an earlier neuron.
Okay, like a cycle to some external neurons. Okay.
Yeah.
All right, thank you.
Sometimes they can even be self loops.
Self loop? What's a self loop? So you mean the neuron connected to itself?
Yeah, I mean the axon, some of the terminals going back to the dendrite of the same neuron.
Oh, I see. Okay, that's it.
Harish?
Hi, this is Bhaskar here.
Yeah.
My question is like, are these neuron, can they change their position or they stay in the same position throughout the life cycle? And the second question is whether this the whole body we have the same type of neuron or different parts of the body have different types of neuron?
Yeah, so neurons stay. So position, well, their size obviously would have grown, and earlier, when I was young, they would say that you don't grow new neurons, but now they say that, well, new neurons can form. So obviously, when they are formed, there would be a change in the size, but once they are formed, they are more or less stable. The connections, there can be changes in the connections, which I'll talk about when we get to learning. But other than that, their size stays the same. They can be repurposed for different purposes, and again, that goes back. We will get to that when we get to learning.
Okay, sir. Thank you.
And sorry, you had one more.
No, those two questions only. One, like the position means if they stay in the same place, and the second one is the means the life cycle.
Yeah. Sumant?
Yeah, hi Omkar. So basically, I just wanted to understand why the axon hill threshold and the myelin sheath were there to avoid what exactly?
So axon threshold, if that threshold is not there, then the neuron will be, firing all the time. So the threshold needs to be there. There are again disorders where the neuron might be, just firing all the time. Like that would be a seizure, like epileptic attacks or things like that, where the neurons are continuously firing without any input. So that would not be good. What was the second question you asked about?
The myelin
sheath. Yeah, myelin sheath increases the speed with which the signal goes through the axon. And again, I think the glial cells which make up the myelin sheath are still being studied by neurobiologists. Most of them thought they were not important, but now looks like they are getting a lot of attention. So the fact is that during human development, they have seen that parts of the brain that develop are also being myelinated at that time. So they suspect that myelination has something important to do with development. And one thing they have seen is that myelination increases the speed with which the signal is propagated down the axon.
Thank you.
Mithun?
Yeah, I had a similar question on the definition of the threshold here, right? So, and how does that basically correlate to the models that we are trying to create in machine learning is something I would like to know. Actually, I probably might be covered later on, but
yeah, we'll cover that. But the threshold and when we talk about learning, we'll see that the threshold also is a variable that could change as we learn.
Right. Get it.
Okay, still a lot of hands up. Let's see, Amit, you just asked your question, or?
I did. I forgot to lower my hand. I will hold it now.
Yeah, Bhimkun?
Yeah, so between this neuron communication, other than aggregating all the signals and sending out the result, is there any other additional like processings or functionalities? Or is it just the signal aggregation and transfer?
Yeah, mainly, just the signal aggregation and transfer.
Okay, thank you.
Yeah. The glial cells, though, they are of different kinds, and again, some of their functionality might not have been fully uncovered. Myelin sheath is made up of one type of glial cells. There are other kinds of glial cells, like I said, which eat away at the terminals that are not being used. And there are other kinds of glial cells as well. So they might have, other roles to play. Lohit?
Hi, Omkar. I mean, if you're covering it later, you can ignore my question, but I wanted to ask, I mean, I understand why we are studying this, but when you said Chat GPT has 1 trillion neurons or 3 had 25 billion
Connections. Yeah,
connections, who thought that mimicking these machine learning in terms of neurons would be more efficient? Like, why did they think like this? Like,
yeah, I will get into that
okay.
later today.
Sure. Yes, sure.
Yeah. Okay, so let's take a break. I was going to take a break half an hour ago, but it's okay. We had a lot of questions, so let's take a 8-minute break. It's 10:57. Let's resume at 11:05 Pacific time. 7 or 8-minute break. Okay, let's continue. Rohit, you had a question. So a couple of you have asked about explaining NP hard, what that means. So it's going to be difficult for me to explain it in a way that you fully understand. But very roughly, I mean, the reason it's difficult to explain is because it requires some theory of computation. In particular, it requires us to understand Turing machines, which are a theoretical model for a computer. NP actually means non-deterministic polynomial, and polynomial, of course, is referring to polynomial time. Non-deterministic is referring to the behavior of that Turing machine, that it can make certain decisions almost magically. That's what non-deterministic means. There's no predictable algorithm that makes it jump to a certain conclusion. It's directly making the best possible conclusion. But in effect, very intuitively, it boils down to saying that the problem is in NP, if I can verify a given solution for it in polynomial time, and I say actually it could be done by a non-deterministic Turing machine. So in other words, if I take a problem that is in NP, say something like the traveling salesman problem, where I want to figure out the best route to take if I want to visit a bunch of cities and come back to my starting point. In what order should I visit the cities? That's a problem in NP. But if I, if you convert that problem, firstly, all of these problems have to be converted into decision problems in the theory of NP completeness. So the answers have to be either yes or no. So a decision version of the traveling salesman problem would be something like, given this map of a bunch of cities and I want to visit n of those cities and come back to the starting point, is it possible to do it with a total cost that is less than or equal to, say, 500 units or whatever the cost is? And if I give you a solution, I say, yes, I can do it. Here's the solution. You can verify that the solution following that particular route can be done in under cost of 500 units in polynomial time. So that's what makes the problem in NP. NP hard is something even more specific. It has to do with problem reduction. So an NP hard problem is a problem into which you can reduce. So I will actually desist from going into that right now because it'll just give rise to more questions. But very intuitively, when we talk about NP complete problems, we're talking about a bunch of problems that are in NP and which can be reduced to each other in polynomial time. And you have to think of all of these problems as decision problems. So if I can solve the traveling salesman problem in polynomial time, I can also reduce the traveling salesman problem to all of the other problems that are NP complete, right? And that reduction can be done in polynomial time, which means I can solve all of those problems in polynomial time, because if I have the solution for every instance of the traveling salesman problem in polynomial time, any other problem that has been reduced to it will also get the solution. Suppose I want to solve some other problem that is also NP complete. I just reduce it to an instance of the traveling salesman problem, which I know how to solve in polynomial time, and that allows me to solve the other problem in polynomial time as well, because every instance of that is reduced to an instance of the traveling salesman problem. So having a solution for this in polynomial time gives me a solution for that in polynomial time, assuming the reduction was also done in polynomial time. And so that makes all of these NP-complete problems cluster together in a way that you can say that if any of them can be solved in polynomial time, then all of them could be solved in polynomial time, because you can all, you can reduce them to each other in polynomial time. I hope that's sufficient for now. But because nobody has been able to build a polynomial time solution for these problems, people suspect that this class of NP problems will never have a solution. So is that good enough for now? Dheeraj and who else first? Harish. Yes, Dheeraj.
Sorry, I haven't raised my hand.
No, you asked that on the chat, right? Q&A.
I think it was another Dheeraj.
Oh, okay.
I have. Does this NP NP problems are anyway related to this connections by any chance?
No. So those have to do with discrete problems, discrete math, computational problems. They are not related to connectionism.
Thank you.
So they have to do with the fact that on a von Neumann architecture, right, a simple machine that can run a bunch of elementary operations, the basic arithmetic and logical operations. How many such, given an input of size n, how long would an algorithm take to run on that input size? So that defines the time complexity of an algorithm, but you can also define the time complexity of a problem that way, if there exists. So the fastest, the complexity of the fastest algorithm on the problem would be the complexity of that problem. And the catch is that you have to be able to identify the problem as polynomial time, even if there is no algorithm that has been designed, polynomial time or non-deterministic polynomial time. So, by the way, every problem that is solvable in polynomial time can also be verified in polynomial time. So, by definition, all problems that are solvable in polynomial time, they're called P, the set P of problems that can be solved in polynomial time. That's a subset of the set MP, which is all problems whose solutions can be verified in polynomial time. So P is a subset of NP, but there is this cluster of NP problems called NP-complete problems, which, if you can solve any of them in polynomial time, would mean that the two sets P and NP would collapse together and they would become equal, just by nature of the fact that an NP-complete problem, if solved, can provide a solution to all other problems in NP. Okay. So, continuing here with the connectionist thread. In 1943, McCullough and Pitts, McCullough was, I think, a neurophysiologist, Pitts was a logician, based on the work done by neurobiologists, which we have seen so far, they decided to mathematically model a neuron, because they were trying to ask the question, could a neuron behave like a logic gate? Right? Pitts was a logician. So if a single neuron could behave like a logic gate, then we could get a network of neurons to implement any circuit or any function, Boolean function that we want. And so they came up with that kind of a model. So they modeled a neuron mathematically as a logic gate, much like how Shannon had built logic gates out of electromechanical switches, and which later people would build out of vacuum tubes and transistors. McCullough and Pitts were thinking of building it out of a neuron. Of course, they didn't physically build it. It was just a mathematical model. So here's an example of the mathematical model of a neuron from their paper. So there are two neurons shown here, neuron 1 and 2. Assume that neuron 1 is firing. or maybe it's not, but assume for now that it's firing. Its output here could be either 0 or 1. Neuron 2 is receiving inputs from neuron 1 only, and you can see 2 axon terminals here from neuron 1 feeding into the dendrites of neuron 2. The output of neuron 2 will have to be a 0 or 1 based on whether the axon hillock is crossed or not. So for all of these examples that I'm showing here, assume that the axon hillock threshold is equal to 2. So in other words, if the sum of all the inputs into the dendrites is greater than or equal to 2, then the output would be a 1, otherwise the output would be a 0. So let's say the axon here sends a signal that is 1 into the dendrites here. So there's 1 here, there's 1 here. 1 plus 1 would get integrated here into 2. 2 is greater than or equal to the axon threshold, which is 2. And so the output would be a 1. Okay, so 1 plus 1 is greater than or equal to the threshold. So the output would be a 1. What if the input was a 0? Then we would have 0 plus 0 making up a 0. 0 would be less than 2. So there would be no output. In other words, the output would be a 0. So basically the output is just going to be whatever the input is, maybe slightly delayed. Does that make sense? That if the input is 0, then 0 plus 0 is less than 2, which means output would be a 0. And if the input is 1, then 1 plus 1 is greater than or equal to 2, which means the output is going to be a 1. So we can even say that the output is just going to be equal to the input, just a delayed version. If you want to show it this way, you can show it that the output at time t is just going to be the input at time t minus 1, slightly delayed. But the delay is not important for our purposes. We'll just assume it's just the same. So it's like the most trivial example of a Macaulay-Pitts neuron. Okay, here is a slightly more complex example. So let's assume that, by the way, the 2 here does not have anything to do with the threshold. This is just neuron 1, neuron 2, the ID of a neuron. So don't get confused by these numbers. This is neuron 1, neuron 2, neuron 3. The threshold, we are focused on neuron 3 here, and we assume that the threshold for its axon hillock is 2. So what will be the output behavior of 3 based on the inputs I1 and I2? We can make a truth table and see what would happen, what the output would be. So that both the inputs could be 0, or we could have any of those other options. If I1 and I2 are both 0, notice that both of them are feeding two axon terminals into neuron 3. So we would have 0, 0, 0, 0 here. All of them would add up to 0. 0 is less than 2, so the output would be a 0. If there is a 0 here and there's a 1 here, then the 1 here will split into 2. So it will be 1 plus 1. And 1 plus 1 is 2. 2 is greater than or equal to 2. So the output would be a 1. By the same reasoning, if this input is a 1, we'll have 1 plus 1 integrated into a 2. So the output will be a 1. And if both of them are 1, then also their sum will be 4. It doesn't matter that 4 is exceeding the input threshold by a bigger amount. The output would still be a 1. It's an all or none phenomenon. So you can say the action potential is a digital phenomenon. Okay, so what kind of logic gate is this? So you can tell from the truth table here that the output is nothing but the OR of the two inputs. To be more specific, you could even write it like this, but doesn't matter. For our purposes, we treat it as a combinational circuit, which means there's no notion of time. It's just an OR gate. Mithun?
Yeah, just a quick question. So the threshold definition that you have mentioned out here, right? So we have, for the first case it is set to 2, and for the second case it's still 2.
Yeah.
Why?
Yeah, it's 2 for all of these toy examples of McColopit's neurons that I'm taking up. There are 3 or 4 examples.
All right, but it can be even having a higher value also, right?
Yeah, it could. So you can define a since it's to be mathematically defined, you can set any threshold you want. Could even be negative or 0.
I see. Okay.
Yeah. Their goal was to elicit the behavior of all of the logic gates, and so they said, let's we can do it this way. Their focus was on a single neuron. Can a single neuron behave like a logic gate? Right? Modeled a single neuron as a logic gate, and the question is, can it be done? At least looks like an OR gate can be simulated by this neuron. I mean, there are 3 neurons drawn in this diagram, but it's really this one we are focused on. These 2 are drawn just to show that the inputs into this neuron are coming from the axons of 2 other neurons. We are not focused on the behavior of these. These will in turn have their own behaviors. But this red box contains a neuron that is implementing an OR gate. That's the point.
Right. So if I then have the threshold set to as 1, say, and like the notion is to kind of get it, simplified into some kind of a logic gates, right? So I can even have an AND gate.
Yeah. So in fact, that will be the next question. How do we build an AND gate using a neuron? And there are multiple ways to build it. Like you said, what if I set the threshold to be, sorry, you said 1?
Yes.
No, that would still make it an OR gate, right? You want both of them to be on. So let's look at this one. What is this? So again, we are focused on this neuron. What is it implementing? Threshold is 2 again.
It's an AND.
Yeah, this is an AND gate. Why? Because we could make a quick truth table again and see that when both of the inputs are 0, they add up to 0 and 0 is less than 2. So the output will be a 0. If either of them is a 1, then there's only a single axon terminal here coming in. So we'll have 1 plus 0, which is 1, and 1 is still less than 2. So the output will continue to be a 0. But if both of them are 1, then they will get integrated into a 2 at the axon hillock, and 2 is greater than or equal to 2. So the output will be a 1. So that means the output is really an AND of the 2 inputs. Right? So that's can be used to implement an AND gate as well. You could have actually done the same thing here. Sorry, it was Mithun, right? Suppose there were 2 axon terminals here. What you try to set the threshold to 1. What's the right threshold to set to make it behave like an AND gate?
I think 4.
4, yeah. When both of them are 1, then you'll have 1, 1, 1, 1. You want to trigger the output to be 1 at that point. So you can set the threshold to be 1. In fact, you can even set it to be 3. Even if you set the threshold to be 3, it would still behave as an AND gate. Because if either of them is 1, the sum at the hillock is going to be a 2. So anything more than 2 is anything more than 2 but less than or equal to 4. So 3 and 4 are the options if we assume that the threshold has to be a whole number. Now I mentioned that some of the connections can be inhibitory as well, because the neurotransmitters released there could be inhibitory neurotransmitters, and they showed this by having an open circle instead of a closed circle. So you can think of an open circle as standing for a value that is to be subtracted instead of added. So what would this neuron be implementing? You can think on your own. I'm just going to draw the truth table and then we'll see. So if this is a 0 and this is a 0 as well, then 0 plus 0 is 0 this is a 0, but then subtracting a 0 is not going to change anything. The output will continue to be a 0 because the summation would be 0. What if I1 is 0 and I2 is 1? Then we have a 0, 0 from here. I2 is 1, so we would effectively be subtracting a 1. So 0 plus 0 would be 0, minus 1 is minus 1. Minus 1 is less than 2, so output is 0. What if it's a 1 and a 0? So if it's a 1, we have 1 plus 1 adding up to a 2. This is a 0, so we subtract a 0. 2 minus 0 is 2. 2 is greater than or equal to 2, so the output would be a 1. And if we have both of them being 1, then we have a 2 here, and then this 1 would have to be subtracted. So 2 minus 1 would be 1. 1 is less than 2, so the output would be a 0. So you can see here from the row in the truth table that's a 1, that we can write the output as a Boolean expression when I1 is 1 and I2 is 0. That's when the output is 1. Actually, what Mikolo and Pitts did is they considered this open circle to be purely inhibitory, which means if this is 1, it's going to block the neuron regardless of what that does. But turns out it doesn't matter, how you define the behavior of an inhibitory connection. You get the same truth table with either of those two semantics. And let's see, what would a NOT gate look like? So we have AND, right? This is an OR gate. This is an AND gate. And there's a zillion ways to build an OR and an AND gate using a neuron, which you just have to define your axon connections appropriately. This looks like, it's not a standard gate, it's first input ANDed with the NOT of the second input. So how about a NOT gate? So if I have a neuron, I want the output to be 0 when the input is 1. So I could take the input. Let's say this input is I want it to be, so what I want is, if my input is 0, my output should be 1, and the converse. So when I is 0, how do I make the output 1? And when I is 1, how do I make the output 0?
You want to change the threshold to -1 or something?
Okay, so let's focus on I being 1 first. If I is 1, I want to make the output 0. So no, if I just make the threshold -1, it will get triggered in both cases. Right, because when I is 0, it's going to sum to 0. 0 is bigger than -1, it's going to get triggered. When I is 1, it's going to sum to 1, it's going to get triggered in that case as well. So
I think the threshold should be 1, and then the I should be subtract instead of
yeah, so let's make it
addition.
inhibitory. I'm just focused on this row for now. When I is 1, I want the output to be 0. So when I is 1, assuming the original Mikolo semantics is going to inhibit it, so the output is clearly 0. When I is 0, I want the output to be 1. So I could set the threshold to be 0. So when I is 0, it means it's not inhibiting. Right? If it's not inhibiting, and if I set the threshold as 0, then the output will automatically be a 1. Because even without any input, it's going to be a 1. And if I use the other semantics where this just gets subtracted, then when I is 0, then subtracting the 0 from nothing is going to give me a 0. So I could use a threshold of 0. It's not the only way to build it, but I could use a threshold of 0. And this neuron would behave like a NOT gate.
It looks like it's not clear. Can I repeat that logic? If I is 0, because your threshold is also 0, subtracting.
So Surendranath, are you making a private comment or?
No, not private comment. I'm asking you, can I repeat it?
Okay, sure. So yeah, so now if you look at, I had focused on this first make it inhibitory, because I said when I is 1, I don't want any output. So I treated it as purely inhibitory, in which case the output would be 0. But even if I use this with the other semantics, that this will have to be subtracted. If I is 1, I will subtract it from what? From 0, right? 0 is the starting point for doing addition or subtraction. So 0 minus 1 would be minus 1. That's what this is would be adding to. So if I have a higher threshold, say 0, the output would be a 0. If I is 0, then I'm subtracting a 0, or alternately I'm not inhibiting it. So I'm just going to look at the other inputs. There are no other inputs. So at the axon hillock, I have a 0. But if my threshold is 0, I could make output as 1.
Subtraction and inhibiting both are different.
Yeah, so there are two different semantics, I think.
Okay. So we can change the behavior, huh?
Yeah, so an inhibitory connection, there are two different semantics you could use. The original Mekolo, its semantics was absolute inhibition. That means it doesn't matter what the other inputs have added up to. If this is on, the output is going to be off. It overrides everything else. Another semantics could be you do a subtraction.
So in the
subtraction
same gate,
at that connection.
So in the same gate, can you have two different behaviors? Depends on the time.
No, it depends on how you define the semantics
okay,
of your
okay.
mathematical model.
Okay, got it.
Yeah.
Thanks.
But the diagram we have drawn both here and our interpretation here, with both semantics, it would be the output would be the same. I mean, it would work with both of those semantics. There might be other diagrams which work with one of them, but not with the other.
Okay, got it. Thank you.
Mithun?
Yeah. So just on the same note, right? So we have the absolute inhibition and the subtraction at that terminal. So if I want to kind of write it in a Boolean algebraic expression, so for the subtraction at that terminal would be 1 minus I.
No, Boolean expression would be based purely on truth tables. Like okay. Shannon would do it, right? You look at the rows that have an output of 1 and you formulate a Boolean expression and then simplify it.
Right. So when you say that subtraction at the terminal, it is that it's like
yeah. I mean, so if this is 1,
right,
I would, when I do the summation at the axon hillock, I would have there's no other input, so it's just going to be 0 minus 1, which is equal to minus 1. That's the result.
Oh, okay.
Now I have to compare the minus 1 with the threshold to decide whether the output is going to be a 1 or not, whether the action potential is triggered or not. So if I set a higher threshold, say 0, then it won't be triggered. So the output will be a 0.
Okay.
Right. So the way I figure out the output is by doing the summation inside the box, but I'm showing it as a truth table only. So the function, the way I write the output as a function of the input will be no different from how Shannon would have written it.
Right.
That's what I did here. I just looked at the truth table to write the output as this function of the input.
All right. Yeah, makes sense. Thank you.
Saurabh? Oh, no question. Vandana?
Yes, hi. I had a question was that what we are keeping on for this gate to be a NOT gate is, one is that we are putting an inhibitory connection there. And secondly, we are putting threshold equal to 0 in one situation and the threshold a different threshold in different situation.
No. This has to be the same in all situations.
Yeah. So it's a 0 threshold when the input is 1. So 1 minus 0 is still minus 1.
No, because this is an inhibitory connection.
Yeah.
So it won't be 1 minus 0. This will, whatever you feed in here, that's going to be negated here. So it's going to be a minus 1.
Okay.
Because it has to be subtracted. So it's going to be a minus 1. And if there had been another input here, that would have been a plus 1.
Yeah. So it becomes minus 1. Then it is still less than the threshold. So it will still give 0. That's what it came. But if it is 0, so 0 minus 0 is still
0. And 0 is greater than or equal to 0.
Okay.
So the output will be triggered in that case.
Okay. Thank you.
Yeah. So it's just by trial and error that I designed this.
Okay.
I just focused on the first row
and
I decided to come up with a threshold of 0 to exceed the minus 1 that I was getting.
Yeah.
And it turned out that it works even in the other case.
Yes. Now I see how it works in other
so
case.
Yeah, so I keep the threshold as 0.
Okay, thank you.
Okay, so since we can implement all of the three standard logic gates using a neuron, it follows that any Boolean function could be implemented as a combination of and, or, and not logic gates, which means a collection of Mikolopit's neurons could implement any Boolean expression. We can implement and, or, and not as neurons. I'm going to just call them Mikolopit neurons. This means we can implement any Boolean function as a combination of logic gates, which in this case are Mikolopit neurons under the hood. Of course, they didn't build this out physically. This would have to be, some kind of separate hardware device if it had to be built out, but they didn't do that. It was just a mathematical model. But you can see the influence that Boole had on Shannon, and Shannon had on Mikolo and Pitts. Even when they were thinking of mathematically modeling neurons, they were still thinking in terms of logic gates. The other thing to note here is there is no learning so far. These are all hardwired connections, hardwired weights. So we are talking about building functions. There's nothing to learn so far. But there is some progress made in understanding how the brain could potentially be working, elucidated by neurobiologists. So one of the line items in McCarthy's 7-point agenda for the conference was neuron nets. The idea was by bringing in people with that background in biology or psychology. Mikolopit's neurons had been already, the paper had already been published at least a decade ago. So attempts to use neuron nets to implement the same functions were obviously known at that time, and so it was part of the agenda. But many of the people coming in from the psychology side were more interested in understanding how the brain works rather than building machines that could automate more and more things that humans could do. So they were not really thinking like engineers, they were thinking more like scientists. Can we construct a model, and a machine would for them just be a model, a mathematical model or a physical model that can perhaps give us insights that we go back and shed and have a better understanding of the brain based on what we see happening inside the model. So it was a very different motivation from what the four pioneers had who wrote the Dartmouth proposal. So how does learning happen? From a biology or a psychology perspective, Donald Hebb made a profound contribution to our understanding. What he proposed is that, and this is the key thing, learning is a change in the strength of synaptic connections, change in the strength of synaptic connection or communication. So when the strength of a synapse changes, and we're talking here about the biology, right, not about the mathematical model. When the strength of a synapse changes, it starts working a little differently. For example, if the strength goes up, then the postsynaptic neuron is going to get excited at that terminal with a stronger signal. Right, so the dendrite gets excited by a stronger signal at that terminal. Now the input that came in from the axon was kind of digital. It's either 0 or 1. But now we are talking about the synapse getting stronger, which means the contribution of that one input to the signal that's sent in, that is triggered at the dendrite has to be more than, say, the strength of other synapses, which maybe didn't change. So this change in the strength of synaptic connections is called, technically it's called LTP, or long-term potentiation, because what they saw is that the strength persists. Right? If you come back after several weeks, you will still see this effect. So Donald Hebb wrote a book called The Organization of Behavior, in which he came up with this new way of understanding learning. He claimed that an increase in synaptic efficacy, or the synaptic strength, arises from a presynaptic cell's repeated and persistent stimulation of a postsynaptic cell. So what makes the strength of a synapse increase? When the axon of neuron A repeatedly triggers the dendrite of neuron B, something happens to automatically strengthen the connection even more. And this change in the strength of a synapse is called synaptic plasticity. And according to him, that's what's happening in the learning process. So here's a quote from his book. Let us assume that the persistence or repetition of a reverberatory activity or trace tends to induce lasting cellular changes that add to its stability. When an axon of cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency as one of the cells firing B is increased. A more catchy way to put it, which was given by someone else, is neurons that fire together wire together. Yeah, but specifically, A needs to take part in firing of B, and if it does so, then the strength between A and B would increase. So how does this happen? Biologically, what is actually going on that could make the synapse become stronger? This is basically what memory would be, by the way. If, let's say, you're mentioning the name of Pythagoras triggers in me the memory of what he did as a religious cult leader, or what contributions he and his followers made to geometry or number theory, it's because the perceptual input of Pythagoras causes some neurons to fire, which, because they are associated with other groups of neurons that are associated with the memory of what the Pythagoreans did in number theory and geometry, would cause those memories to also surface, because those neurons are also firing. So there are associations in my mind between different things that Pythagoras did, and so all of those associations get triggered by the triggering of the the neurons that respond to the name Pythagoras. So how does this happen? So there are three mechanisms. There are three mechanisms by which the synapse can get strengthened. The first is that there are receptors inside neuron B. So when I say A and B, A is the presynaptic neuron and B is the postsynaptic neuron. So keep that vocabulary in mind. There are spare receptors in neuron B. Spare receptors in neuron B that are not placed on the cell membrane, on the dendrite membrane. They're just inside the cell, and they get put they get put onto the surface of that neuron. So if I increase the number of receptors on the surface of neuron B, then the same neurotransmitters, the same concentration of neurotransmitters that are sent, they will now have more receptors to bind with. And so they will bind with those receptors together, and there'll be like more, a stronger signal that will be sent, right? Because now I have 10 receptors that are all getting triggered at the same time instead of 2 or 3. So when A fires B, and this happens relatively frequently, there is a complex biochemical pathway which can cause this phenomenon to happen. More receptors being put on the surface of B. The second mechanism is that there are neurotransmitters that can be secreted by B back to A. These are called retrograde neurotransmitters. They are special, I mentioned there are 100 different types of neurotransmitters, so a few of them would be retrograde. These are sent from B to A, and what they do when they reach A is they cause A to increase the production of the regular neurotransmitter. So increases the production of, I'm just going to mention the name of the neurotransmitter, glutamate, right? The neurotransmitter associated with learning, the normal neurotransmitter, or one of them associated with learning. So a cell like A can manufacture more neurotransmitters pretty easily. Neurotransmitters they have simple precursors, simple amino acids or lipids which are there in our regular food. They can be manufactured on the fly and relatively quickly when the need arises. And so these retrograde neurotransmitters can increase the concentration of those neurotransmitters that are actually going to go across from A to B and fire, or try to fire B. Right? I'm talking about this concentration of glutamate here. If there's more glutamate manufactured in A, then there's going to be you'll see more of these chemicals across. And if there are more receptors here, they're going to bind with more of them. So the signal that will be sent from here is going to be much stronger than what it was before. Not just that, the shape of the dendritic spine here can also change so that the signal is less attenuated by the time it reaches the axon hillock. That's the third mechanism. The third mechanism is that the shape of the dendritic spine in B, when I say spine, I'm referring to the fact that cells also have a skeleton called a cytoskeleton made of certain protein fibers that maintain the shape of the cell. And so if I locally dismantle the skeleton of the cell and rebuild those fibers in a different way, the shape of the cell can change locally. And that's what we're talking about here. The shape of the dendritic spine in B changes to allow for the signal from A to sweep or to reach further, to sweep further without loss of concentration or intensity, or whatever you want to call that, whatever way you want to measure the magnitude of that signal. So it's more likely to reach the axon hillock with a bigger value. So a combination of all of these effectively increases the contribution of a particular input at the axon hillock. And this is what points us to the meaning of the word connectionism. Connectionism refers to the idea that knowledge, whatever knowledge we have learned, is stored in connections or synapses, synaptic strings, you can say, between neurons. So when you learn something, it's not like new synapses are forming or that new neurons are forming, the connections between some of those neurons are getting strengthened. So this is the idea behind connectionism and the alternate approach to AI that we're going to look at next. So let me just compare and contrast what two approaches we are talking about before I go back to our mathematical model and change and enhance it to incorporate learning as well. Actually, maybe I should ask you, what is the simplest way in which we could change our mathematical model to incorporate this known mechanism for learning? Okay, Mithun?
Well, I had a question on the other part, but to answer your question, it could be a simple feedback loop.
So suppose this is my neuron, and so far what I had was I had inputs x1 to xn. They were either 0 or 1. The output was 0 or 1. I had a threshold t, and my semantics was that x1 plus x2, if I add all of those, this becomes t, sorry, greater than or equal to t, then the output is 1, else output is 0. What can I change in this model to incorporate the fact that we have synaptic connections that are getting strengthened, and let's say one of these connections is strengthened? I should be able to have this connection contribute more to the summation here.
I would add weights to these.
Yeah. So the simplest way would be to add weights to each connection, indicating how strong they are. So now we have the following changed or enhanced semantics. If x1 times w1 plus x2 times w2 all the way till xn times wn is greater than or equal to t, then output is 1, else output is 0.
Ultimately, the result is the same, right? Whether you add weights or not.
No, the result could be different.
1 or 0, output 1 or 0.
Yeah, but which new neurons? If you have this is a single neuron, yeah, the output will be 0 or 1. But when you have a collection of them arranged into a network, then which of them gets triggered and in what way will differ after learning has happened.
Got it.
Than say before. So just to take an example, suppose t is 7. Right? Suppose t is equal to 7, and let's say the weights are, I'm just putting some random numbers here, 5. Let's make this inhibitory, -3. Let's say this weight hardly contributes anything, it's 0. This is 1, and maybe this contributes 10. Right? If all inputs are 1, then what will the output be?
5 minus 3, 0, 1, 10.
Yeah, so we have 1 times 5 plus 1 times -3 plus 1 times 0 plus 1 times 1 plus 1 times 10. So that's 5 minus 3, which is 2, plus 1 is 3, plus 10 is 13. Now 13 is greater than or equal to 7. So the output would be a 1. This neuron will fire.
So one quick question I had is like, then how are you modeling that strengthening? You said like the weight, right? So in this case, we are having an arbitrary weight assigned.
Yeah. So what should happen during learning is, so learning should change the weights. That's all that learning should do.
Okay, so which means based on the output is 1, some weight is going to change, like output weight, maybe 5.
Oh, so we still have to figure out.
Okay.
How to make this, how to build a network that could make you learn something. But so far we are just talking about the biological mechanism for learning, based on which we are enhancing our mathematical neuron to incorporate weights to reflect the strength of synaptic connections. Now, how we are going to use this, that's going to come next.
Okay, thank you.
But this is the requirement, right? If we have to be relatively faithful to the biology, then learning should be about changing these weights. And by changing these weights for all the neurons, potentially for all the neurons in the brain, we should get new knowledge. So Rahul, question?
Yeah, so my question is like, we have been talking about negative thresholds, negative weights, negative inputs, and everything we are talking in terms of strength. So what does negative strength means? Like, I understand, you can increase signal strength to 1, to 2, to 3.
Yeah, negative strength means this is an inhibitory connection, right? So the neurotransmitters for this connection are inhibitory neurotransmitters. So instead of helping, instead of contributing a positive amount to the summation, they will contribute a negative amount. That's what a negative weight here means.
That I understood. Like, it is decreasing it, but physically, how would you denote it? Like, logically, I understand that, but physically, how is it denoted?
I mean, physically, there would be some biochemical pathway triggered here, which would go and block the signals or block the flow of information from other dendritic units to some degree.
Right. So blocking means making it 0, but then making it negative, that's like a bit counterintuitive to.
No, so there will be some chemicals released here. Some chemicals released here, which will block other chemicals that could have been released there. And if there's nothing being released there, then these these inhibitory chemicals will dominate at the axon hillock. But in either case, there's going to be no action potential unless you have a positive signal here exceeding the hillock. So we are just talking about degrees now. We are just talking about, it's not a digital phenomenon. And so these weights are reflecting the fact that it's not a purely like an all or none kind of analysis here. There are weights now.
Right. Now, like talking in terms of, let's say someone is building a hardware to showcase the same thing, right? Sometimes someone must have built some hardware, right, to mimic this. Then again, negative strength, how would that be replicated?
So obviously, the way people did it is mainly by simulating this on a computer rather than building a separate piece of hardware to do this. Although that's what Rosenblatt, who I'm going to talk about, he did try to build hardware that could, that was, so he tried to simulate this on a computer, but computers were too slow in those days. So he tried to also build his own hardware, build his own machine that could directly, execute this. And so it would be some kind of analog device. I don't know what that hardware looked like, but he did build an analog device physically instantiating a neuron unit like this. Of course, it was an artificial neuron, not a real neuron. But so I haven't studied analog devices, but people were in those days building devices that could even solve differential equations using analog circuitry. So Vannevar Bush, for example, I don't know how he built this, but there was a machine called a differential analyzer he had built for solving differential equations. That was, an analog device. And Shannon was working with him on that. And then when he went for his summer internship, he got the idea of using Boolean algebra for improving circuit design. But otherwise, the actual work he was doing till then was on, working with Vannevar Bush on his differential analyzer. This was supposed to be like one of the most complex analog machines until the digital computer was invented. And once you have a general programmable computer, then the utility of specialized analog devices becomes less, because now you can just simulate a bunch of artificial neurons on a digital machine. You don't have to build a device that actually behaves like this.
Very interesting. Like all this, since it's like start from the basics of everything, it's all very intriguing. And eventually, when you explain the concept, the end concept, you're able to like understand it very well, since you know all the basics of how we get there. Thank you for doing that.
Yeah, thanks. Mithun?
Yeah, can you just go back to the biological slide, just the slide before? Yeah. So not
this one.
Yeah, this one. So when you mentioned the retrograde transmitters, so these are like kind of the transmitters, neurotransmitters that are kind of sending the signal from B to A. Okay, and so that is a kind of like a feedback, right? What I understand.
Yeah. So that's what I meant. I think, I don't know if you had asked about that earlier, but I said, I'll come back to this. Yeah, this is a kind of feedback, but it's not the kind of feedback that triggers an action potential in A. It's just stimulating A to produce more neurotransmitters at the axon terminal with B.
All right, and okay. And what about the third point? Like you said, the shape of the spine, right, in B changes to allow for signals from A to sweep further. So what do you mean by sweep further? Is this like...
So the strength of a signal, you can think of it as maybe a concentration of ions that are, traveling. Suppose I have a bunch of ions that are moving, and if the shape of the cell is wider, maybe they can move they just get scattered in all directions. But maybe I change the shape of the path, the skeleton, so that they're all moving in a direction towards the hillock.
I see. Okay.
So that way, they reach there with a higher concentration.
Right. And the last question is about like the basic summarization of this thing is like to retention, right? Like retaining or storing or having a knowledge of some previous kind of an input, right? So my question here is like, by having these connections, like having multiple connections between two neurons, we're kind of then visualizing it like, okay, it is able to retain some previous information. Is that where we are trying to move ahead? Like
yeah, long-term potentiation means that. So we are that in whatever information we have learned is now stored in a bunch of synaptic connections, so that if we get a cue, CUE, or a hint about, or quiz about whatever that concept is, that would trigger a set of neurons, which, because they have strong synaptic connections with other neurons that store other pieces of information about that concept, would make that knowledge surface. So it's like retrieval, right? When you practice retrieval for any exam, you quiz yourself with, you don't directly ask about the thing that you're trying to remember. You solve problems that are sort of around it, right? So I mean, imaginatively, these are all neurons that are sort of surrounding the cluster of neurons which actually store the concept we are interested in. But by practicing the stimulation of those other neurons and the connections to the cluster of neurons that actually surface the concept, we find, or we build different pathways to recall knowledge of or retrieve knowledge of that concept. So the more we integrate information about different concepts together through these stronger connections, the more we sort of enable ourselves to retrieve knowledge of that concept.
Yeah, makes sense. Thank you.
This is obviously all still under research, so a lot of this is going to feel hand wavy. I mean, a lot of what I'm saying is hand wavy. So Rosenblatt was not part of the Dartmouth conference. He was a psychologist at Cornell University, but he was influenced by this work on neuron nets. And so he decided to, like I said, build a network of neurons to solve a problem that McCarthy and others were not emphasizing when they came up with this notion of artificial intelligence. Because like I said, artificial intelligence, that phrase seems to imply that simple unconscious tasks like perception or reaching under the table to pick up something those are not intelligent tasks. But getting a machine to do those, particularly trying to use a logic-based AI approach to make a machine recognize, say, a character written on a piece of paper, uh, or a face or an animal. It's not clear at all how to get a machine to do it, because humans themselves don't seem to do it consciously. It's just something that happens in the unconscious, and we just recognize the face. It's not like we are using rules of inference to figure out who it is, who that person is before us. And so he went in a different direction. And let me just write down the summary of those two approaches. So you can say a twofold approach to AI. The first approach, the logic-based approach, was called GoFi, good old-fashioned artificial intelligence. And the contrasting approach was connectionism. And they were both built around different assumptions about what intelligence is. The GoFi camp of researchers, which included McCarthy, Minsky, Newell and Simon and others, felt that the core of intelligence is symbol processing. So if you want to build a machine that's intelligent, then we have to program it to follow those rules of symbol manipulation. So programming the machine to follow explicit symbol manipulation rules is what will give it new intelligence, will give it new capabilities. Whereas an alternate approach would be to not try to directly mimic what we think are rules of symbol manipulation. We don't focus on what intelligent people do when they're solving problems. Instead, we go down to the root of what's going on in the brain. Since the human brain is the only intelligent, or that's the reference for where intelligence comes from, is the real intelligent thing we know, or source of intelligence. Let me write it in. Since the human brain is the source of intelligence, what should we do to make a machine intelligent? To make a machine intelligent, make it simulate the way the human brain works. Works and learns. This is the idea behind connectionism. And the AI community was split between these two camps. They were often conflicting with each other, fighting for funding. So Rosenblatt was, like I said, a psychologist, and psychologists tended to favor the connectionist approach for obvious reasons. So he used the ideas of Donald Hebb on what it means to learn something. He had already, for example, he incorporated the fact that there are weights that need to be added to the mathematical model of a neuron. And by the way, if you add weights to the mathematical model of a neuron, does that change how you, does that reduce the power of a neuron in any way? It shouldn't. Right? You should still be able to implement your logic gates. I want to do that because it might seem a little repetitive, but I want to use it to also show visually what's going on in a neuron unit. Just like, algebraic equations can be visualized geometrically. We want to come up with a way to visualize what a single neuron unit is doing, because that's going to be crucial in understanding how Rosenblatt's neural network is going to work.